Un, deux.
Okay. 1, 2, 3. Yes. Okay.
Great.
We're set. I guess you will want this.
Yeah, we have
you
others
have a-
here, but
Might
yeah.
as well use this
Yeah,
one
I
unless
can just hand it over.
because you never know.
Yeah.
And we have another 10 minutes. So
Oh yeah.
we can all just
10
chill
past,
here.
yeah. Yes. You came, hello.
Hello, I'm Valerio from
Hello.
the mail. Valerio Laghi from the
Valerio.
mail for the interview.
From
The mail for the interview for MIFOBIO.
oh yeah, of course. Happy to comply.
Whenever you want. So
Yeah.
I was wondering if you know my window for Wednesday.
Whenever you want to tell me, or we can do it tonight. Whenever
Okay,
you
we
tell
can
me.
do it even after your talk.
Yeah.
If you want, we
Yeah.
can go a little bit outside.
Yeah.
Okay.
Super Camille? No, what was it? Valentin. Valerio. Valerio. Di dove? Roma. I pretend to speak French, Italian, everything.
Because you didn't have much of the accent in that sentence. Okay. Maybe.
I had an Italian teacher for the family class.
Yeah.
Maria
It seems
Pia
to be stable.
Garibaldi di Milano.
Okay. Oh.
She was very tough.
Wow.
And when she
Oh,
asked,
it's
she
a
wanted
Mac.
to
Yeah.
so I studied very hard. Hello.
You're Max, right?
Yes, yeah.
Hi,
Hi.
nice to meet you.
Nice to meet you too.
Clement, he recruited you,
Ah,
right?
yeah, he recruited me. Yes, yeah,
Nice,
yeah.
nice, okay, okay.
No,
Yes, yes.
Everything
very,
good?
my
Yeah,
part
yeah.
is,
This
very
the
good
laptop
for
seems
the
to be
whole
more
embedded
stable than
Jasper
yesterday.
lab.
Good, yeah.
So it's
So
okay.
I have visited
But
it
That's a
many
success for a
times.
Mac, yeah.
Yeah.
They've been on one
But she
week
said already she has
classes in
uploaded it on
Geneva
Google
and-
Drive, so in case we can switch.
Ah, okay, okay.
They
good.
start like
Yeah,
crumbling
yeah.
again.
But always well, Eric no, but the rest is the north.
Yeah,
Okay.
yeah, his group is in Genova, so
Oh, nice.
he's a fantastic figure in microscopy. He's funded.
Deux,
There
un.
are so many students from
Ah, mais je sais pourquoi. Voilà. Donc, il fallait activer les deux pistes. Il n'y a pas de problème. Hein, c'est pour ça qu'on va jouer la gauche. Mais bien sûr.
Non, on peut pas avec les...
Un, deux, test. Un.
Leuven is not very big.
No.
Bah voilà.
Yes, actually, I'm working in the group where he was with Peter Delek.
Oh yeah. Yes.
Nice place.
Santa Cruz. I'm really jet lagged.
I can imagine.
I wake up in the middle of the night. It's like, what am I gonna do now? Go out and watch the stars.
I went for 2 weeks and an internship in San Diego.
Oh, where
And
at?
yes.
At the company or?
No, it was a
This is
hospital. It was a
Hospital.
Yeah, I don't remember. Oh, the name was Perdido. I really don't remember totally. But there was this group that worked on and I remember the first week I didn't manage to sleep a normal hour.
Yeah, it's pretty.
When did you arrive?
A couple of days ago I went to sleep.
Okay. Okay, yes,
Yeah.
yes. Okay, see that.
Okay, we're getting rest here. Maybe we can sit here.
Yeah, still have 5 minutes, no?
Yes.
Yes, yes.
Okay.
This is the least COVID good for
Yeah.
the whole thing, right? Because at least at
Yes.
dinner we are spread out a little bit, but here and the air gets so bad. I walked in here late one night
Yeah.
and like,
Yes.
it smells like a gym in here.
Exactly. Yeah.
No, but I think it's even the humidity from
Yeah,
the
it's the
sea that
humidity and
blocking here.
yeah.
Because I think in summer there are like spectacles, no? There are some
Yeah, probably
some
animation.
program animations for families because it's very popular destination in France.
Really?
Yeah.
I
For,
can believe it because it's
yeah.
beautiful.
And it has to be beautiful in summer.
Yeah. Yeah, I was looking at the beaches. Like, I wonder how cold this is.
Well, there are people who were swimming yesterday
Yes,
who went
yeah.
swimming, so
It's
you
not
can
as
try.
cold as you would
Yeah,
suspect.
Try it.
It can't be worse than the Pacific Ocean in the north of
No, I
California
don't
or
think
the
so.
Baltic Sea.
I don't think so, no.
If you try it, the jet lag will no more be a problem.
I think
because
you're
you
right.
will get
Yeah.
really awake.
I would
really
be whenever I feel tired. But there's also a swimming pool here. Has anyone been to it?
There is a swimming
Yeah, there is a
pool,
swimming
Where
but
is it?
I know there is,
think
but
it's
I'm
warm.
You know, if this
It's
would
okay.
be the 1990s, all the students would be swimming in it at night.
Yeah.
Safe, not-
Yes, we have experience with that. I think it's somewhere more further, probably.
No, it's not.
The swimming
it's
pool?
about the
Yeah.
little housing complex. Because
Yes.
I looked through the trees and I see it. So
Oh,
it's
Yeah, it's
here.
very close.
Okay,
Yeah,
Close
yeah.
to this club.
Yes, yes.
So-
that's
Yeah,
true.
it's
So
Yes.
why we didn't organize our
this
party
is
of
what
the...
And you will
Full
heat
fight.
the water for the people?
Someone
Just a
will,
little
after
bit of-
alcohol,
yeah,
someone will
so
fall
the,
into water.
Yeah, someone will have to
Liquid
do the
sweater.
best.
Yeah.
Anyway, will be the last day. So if someone die the day after, it's no more our fault.
It's not your responsibility, you want to say?
I think this is, yes.
Oh, I mean, we can check if you want.
We can try to organize that. I don't think they will give us the clearance because there will be someone to check.
But there is probably not a lot of staying now, are there?
Till Thursday party?
I mean, us are staying, but there's not so many guests. Should be.
No, it's empty.
Yeah, so
Really
it's just
empty.
us.
Empty no, because I saw some people
Not
walking.
But there are
completely
people walking past that have nothing to do with this. Because some girl was
Yeah.
asking me for my cell phone, and then her boyfriend was there screaming at something. It was really strange.
Oh, really?
Yeah.
No, it's not completely
Maybe I'm
empty,
too
but-
American and it was totally fine, and I'm just seeing danger everywhere.
No, it
Because if
was.
this happened
if you-
in the US, I would go, okay,
So
sure
you were
you'll find one
working-
at the reception.
you were working, she came to.
She came up to me,
No, that's strange.
and then later I saw her with his boyfriend, and they were screaming at each other. And
Ah, okay, that's a bit
okay.
weird. Yeah.
it's,
No, but otherwise the complex is almost empty, let's say.
yeah, so
Yeah,
you guys
yeah.
can have a quick party.
Because I think the season is over, right? I think
Yeah.
they're already
Yeah.
over
November,
till-
yes.
So in November there are really like not many people, or even it's closed, I don't know.
So without us, they would be already on holiday.
Yes. Yeah.
No, actually no. They will have another conference.
Yeah, they will have
All these
so
places
the
divided
stuff-
by conference.
There will be
probably
No,
all
they
the
close-
time
they close after us.
this winter. Otherwise, it's always.
They will
Yeah,
close after us.
That's true, they do.
I mean, they have all the equipment.
But they should
Because
keep
for
them.
example, I know some
Yes, it's a bit
resort
cold.
in Italy that
Yeah.
is in Sardinia.
I-
They remain open
there's
during
like
the
a
winter
heating
because
thing
they do
in my
one
room.
conference
Ah, yeah.
after the other.
And there are
Till
radiators.
they start with
Try
summer.
to say now I'm sleeping like in the 3 layers.
Probably it will be different here because it's not really.
Yeah, maybe the changing season.
Basically, for this,
Not yet really winter.
November is winter, no?
It's winter, switch it on yet. But it's already a bit cold. At night at least.
Well, I discovered that you can put air conditioning to hot here.
I tried that, but I don't
It
think
doesn't
the air
work.
comes out as warm.
If you put at 25, mine
I
is
put
like
at 30
fire.
actually.
For mine was like fire at one point.
Really? I also put it on 25
You,
minutes.
you have to go
Okay,
to
I'll try 25.
mode.
Yes?
You click on mode, you have the little sun appearing. Then you put on 25.
I have it on the wall.
Yeah, but on the
Yeah.
remote control.
The remote control there is mode,
Yes.
and you, if you check, go from winter snowflake to sun. Then you wait for 30 minutes and start to
30 minutes. Okay, I
try
can
this.
wait 30 minutes.
This is a good
20 minutes
engineering.
depend. Depends by your.
just left it alone so far.
What?
Okay. I just left it alone, and
Yes, but if you leave, you take your badge and there's no electricity.
Yeah, you have to put in another
So it
credit
will not
actually,
work.
if I leave,
Yeah, you put
there
in a
is.
credit card. Into the
You can put
Actually, I
whatever.
don't know, but mine works without the card. I don't know why.
Wow,
It does,
you're in
yes.
a nice room, huh?
Or you just broke something. You didn't know.
It does blink, like the LED blinks, but I don't.
I usually put directly credit card, a card from Carrefour inside,
Some
something like
other
that.
card,
yeah.
yes, it will work also.
But I still don't have a roommate. I think I will never have one. Got lost.
That's good. For you have your bathroom only for you.
When I lived in New York, I had a roommate who was from Belgium, but she was a medical doctor. She worked on immune things, and she said the great things about the whole medical system in Belgium. They sort of
This is very great. I have to admit I really like it.
Yeah,
It's very
what I
Roman
hear, the whole
Republic,
Dutch one is terrible.
Really?
The Dutch one is supposed to be terrible,
This I
and
don't
they
know.
are so close to each other.
Yes.
But
Okay.
somehow it's a completely different country.
Yes.
Yeah.
Well, the Belgian one I really like,
Yeah.
I have to say, and even for like paying the Dutch one, the reimbursement by the insurance company, insurance service,
Yeah.
is really fast. I'm surprised.
No, I didn't know when I arrived in France it was half private, half public.
France is supposed to be very good too.
Yeah, but it's like
Sweden is falling apart and it's horrible.
No, but I feel the Belgian one is a little bit better than the French one,
Even better.
because of the
Yeah, that's what I
administration
was
and the
wondering.
delay and stuff.
then you need to call.
Because our doctors say that France is the best healthcare system in Europe, but that's because Belgium is so bad.
You mean your doctor?
I will start before you for small communication. Just
Alright.
Okay, then we have set up my presentation. I hope it will work again when we reconnect it. Okay, very good.
I can always say because in France is a half, most, you have a private part and a public part.
Yes.
While I come from Italy where it's everything public. So I didn't know at the beginning that I need another well, somebody if you want. Huh? You don't need it. Well, but it stopped
if you
before.
think you may need it.
Yeah. It stopped before you have some kind of need for the
It depends on your risk. If you're, I don't know, you need optician a lot, you have need for.
or if you need a surgery, they will make you pay, for example, the anesthetics, the room, yeah, while in Italy it's all public, you pay 50 euros
It's both. It's just sort of like that. So there is Medicaid
from the center.
for anyone who doesn't have money. And then if
The
you
problem, the other
have
flip of the coin is that in Italy it's all public, so
and
there are
that's
really good
having
places
insurance
and
is
really
the
bad
best
places.
option.
Sure, yeah.
I'm sure it's maybe even in line with Belgium. Like
I
if
can
you get
believe that, I don't know.
yeah, if you have good health insurance in the US, it's really good.
Isn't it difficult to obtain the health insurance, or it comes with
It comes with your job.
Okay.
And if you don't have it, if you have no money, then you have what's called Medicaid, which is basically the yeah. Yeah. So it's the same system. You have a private version, which is a little nicer, and then you have like a basic Medicaid. But a lot of it is based on going to the emergency room if you don't have insurance, which makes the emergency room worse. However, in the US, when you need to go to the emergency room, it's no problem.
Okay.
Because this is where everyone who doesn't have insurance goes.
Ah,
And they cannot refuse it.
yeah, sure,
If you go to
yeah,
the ER, they can tell you like, oh, sorry, you're not sick enough.
Why Sweden is not public?
Sweden is all public. They used to be all public, but it got so bad that they had to allow private healthcare because it was not even legal. And now it's much better, and healthcare personnel. But we have a crisis. Right last week, 150 of the midwives and obstetricians quit because women's care, of course, is worse than any other care. And it's so bad in the maternity department that they quit in process.
Not expensive.
Our politicians have ruined a very good healthcare system. It used to be very good, and it's still you can get very good quality care, but with research but you cannot get it.
You have good alcohol.
What? You have?
Good alcohol, I remember because
Yeah,
when I was in Norway, I took the bus just to go to buy it in Sweden and then come back.
which is bizarre because it is really expensive in Sweden too, but it's even more expensive.
3 hours and a half of bus to go to come back. Rasmus is
At some
on Trondheim and KNU.
that's very expensive.
Yes, I say let's go somewhere where you actually do your job instead of paying. Because at the beginning they told me, do you want to go to Spain or Portugal? And I would never do anything in Spain.
This is how you in the European Parliament you see that there are Italians, but Italians who do studies now.
Yeah.
The center of yes, so we're
Well, alright, thanks for coming and
We go sitting here. And,
Ok. 1, 2, 1.
Bonsoir, j'espère que vous êtes... je sais pas si Okay. Thank you. Thank you, sir. Ok. Bonsoir, j'espère que vous n'êtes pas trop fatigués après cette journée intense, cette deuxième journée complète. Donc rapidement, je voulais faire une petite communication, remercier à nouveau nos partenaires, vous indiquer que probablement on va faire la photo collective mardi en fin de matinée. Donc il faut qu'on vérifie avec les activités sportives pour que vous soyez rentrés pour midi. Et dans ce cas-là, on la ferait à ce moment-là, devant le Bélambra. Ensuite, c'était pour une petite communication. Comme je vous l'ai dit au départ, on a souhaité, dans MIFOBIO, avoir une action de développement durable associée à MIFOBIO. C'est quelque chose qu'on voudrait inscrire de manière durable aussi dans les prochains MIFOBIO. On a choisi une action qui consiste à la restauration de la forêt des morts, des massifs des morts, qui a subi un gros incendie l'été dernier, avec entre autres des tortues Hermann qui sont protégées et qui ont subi cet incendie. Par rapport à ça, on a donc lancé une action avec l'ONF. On va avoir une intervention demain, lundi, à 12h, d'un intervenant de l'ONF, pendant un petit quart d'heure, puis après, ceux qui veulent pourront discuter avec lui sur la stratégie de reboisement de ce massif. Et on vous sollicite individuellement. Donc on va le faire nous aussi au niveau de l'ensemble de l'école MIFOBIO et du GDR, de faire un financement sur ce don, enfin faire un don, pardon, pour ce projet de reboisement. Et on vous invite tous collectivement à participer à cette action en allant faire un don sur ce lien, ou alors en flashant le QR code qui est dans le couloir, un peu partout. Et si vous pouvez penser à mettre MIFOBIO, MIFOBIO dans votre adresse, de sorte qu'on puisse après demander à l'ENF qui nous fasse un peu le total et que je puisse vous dire combien l'ensemble de l'action MIFOBIO, de combien on aura contribué et combien d'arbres on pourra replanter. Donc maintenant, on va continuer avec le programme scientifique et avec les deux, le séminaire et la plénière du module de ce matin qui a été déplacé en raison des décalages horaires pour permettre aux collègues d'être à des heures décentes. Voilà. Donc je vous remercie. Thank you, Sarah.
Hello everyone and welcome to the seminar talk. I'm happy to introduce our next speaker, Sarah Abrahamson. Sarah is an assistant professor at University of California, Santa Cruz, and her main research aim is aiming at the development of novel microscopy techniques, in particular for 3D bioimaging. And we're going to hear today about one of those techniques called multifocus microscopy, and how to achieve 3D super resolution imaging by combination with structured illumination microscopy.
Thank you, Max. I am so glad to be here. I just want to say that it was supposed to happen 2 years ago. And between that, we've had a very hard time in the world. And this is the first meeting in person that I am at after the whole pandemic and lockdown and everything. So I'm very, very glad to see all you and all of old colleagues, new and old. And today I will tell you a little bit about the imaging that we have developed in my lab and also of my PhD work. So I started out when I was 25 and I came from Stockholm to San Francisco. And there I met Mats Gustafsson, who was a very, very new professor, assistant professor at UCSF, and I was a very new master student in physics. And he tricked me to do a PhD with him, and there I met these fantastic people, Lin Xiao, Hesper Rego, and Reto Fiolka, who were the Gustafsson lab. And this was a really exciting time in microscopy, because this is when the super resolution started to happen. So for all of these years that microscopy has existed, which is hundreds, the last 100 years, the objectives have developed and become really, really high NA, and we can now image with really great resolution. But up until maybe 20, 30 years ago, this was limited by diffraction. And then some people started to find ways to sneak around this diffraction limit, and Mats was one of them. And this is Lin's data set, which is one of the first 3D live imaging of biological samples. The color is encoding depth, and what you see is mitochondria. And we all know that in a microscope we can see cells, and we can see the subcellular structures like the mitochondria and the nucleus. But now with these techniques we can start to look inside the organelles, and we can start to do this live and maybe even in 3D. This movie was recorded at 20 seconds per frame, and now, 14 years later, or 13, we can reverse that. So now it's 20 frames per second. So I wanted to talk a little bit about how that come to happen. And what my lab does, really, I'm an optics geek, and that's what I do. I'm interested in the optics, so if you ask me about biology, it will be really fun and embarrassing for me. So please do that later. So we do a lot of nanofabrication of our own devices, we build systems, and we have a lot of fun developing these new optical techniques. So in order to talk about 3D imaging, I want to start to talk about 2D imaging, because 2D imaging is almost all the imaging we do. We're humans, so we're a little cooler than cameras, so we actually have stereo vision, right? So we are almost doing a little bit of 3D information here through the parallax. But if we think about photography, this is a 2D image, and that's the basics of imaging. And we who are microscopists, we think about numerical aperture, and those of us who are also photographers, we think about F number. So F number was one of the things that the F64 group was pressing in the straight photography movement, when you really start to try to image the world as it is. And it turns out that if you close down the aperture of your camera to an incredibly small pinhole, you get almost infinite depth of focus, and this is what they used. Of course, in microscopy, we do the opposite. But if we think about it, we have all the 3D information here, from the shores near us to the mountains away from us. This is the High Sierra. And thinking about that for everyone here who's a scientist, artists influenced the world too. It was artists who started this whole photography movement that saved a lot of the environment in California and saved the whole Yosemite Valley from being dammed. So here we have an open aperture, and we start to distinguish 3D information by destroying the autofocus light. So this is what happens when we open up our aperture, and this is the type of imaging we do in microscopy all the time, right? We try to destroy our depth of focus and get really high resolution in Z. And we can scan through a sample like this, and this is how we normally take a focus stack, right? We sequentially go through the sample, and then we get this focus stack. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, and we can keep going, but 9 planes is this example here of a focus stack. Now, when we do that, in this case it's a flower, so it's not moving very much, but some things really move. Living things tend to move a lot. Even water moves, and this is my PhD student Eduardo illustrating temporal resolution on a waterfall in Cataratas de Amador in Peru. So we know we need high temporal resolution, and we know that today, this is Harold Edgerton's photography from back in the time when you shot with film, and he wanted to have really fast imaging, so he knew that he cannot keep exposing and rolling back his film, or even if he's filming it with a video, it's really not as fast as he wanted to shoot this serve at the Wimbledon in, I think, 1947. So instead he strobed the light to capture this on a single image frame. And there are all these tricks in microscopy, and what I did for my PhD was really one of those tricks, and that was trying to find a way to record this whole focal stack in one shot, so that we don't have to sequentially image in 3D. Because when we scan through the sample, even with a fast piezo stage, we have an ambiguity in our volume in time. And it turned out, when I started out as a PhD student, that people had already kind of tried to do this. So one was Raymond Ober, who took a prism and put into his light path, and then he refocused one of the images. So he had 2 cameras and a prism and a little bit of refocus. And then you already have 2 simultaneous focal planes. You don't lose any resolution. Of course, you lose half of the signal in each image, but that you would do if you scanned as well. But then there was this other guy, Alan Greenaway in Scotland, who had thought that diffractive optics are really, really cool. And maybe if you put an off-center Fresnel lens in a system, what should happen would be that we get sort of multiple focal planes at the same time. And Mats thought this was genius, and he said, this guy is really cool. And I actually went to Scotland. He lives in this stone house in the Scottish Highlands. And I talked to him about this, and he said, yeah, but it doesn't work because, chromatic correction and the aberrations and all of this stuff. So I said, well, if we do a few things, maybe we could make it perfect. He said, yeah, okay, maybe you could, go ahead. So Mats told me that, this was my PhD project. Why don't you build the perfect multi-focus imaging system? And you use diffractive optics, because with diffractive optics you can do almost anything with light. You can multiplex your beam, you can completely control the phase of the light, which means that you can introduce a focus shift with complete control. So that was my task. And it was really, really fun, and I spent many years on it. I'm thinking that most people in this audience already understands a lot of diffraction, but I just wanted to bring it up in case someone is a super expert on biology and didn't really remember Young's slit experiment, that light really ripples like water. So we have diffraction, we have, everyone knows in our eye we have refraction, and then we have reflection in a mirror, which we use in telescopes, but this is diffractive optics imaging. So if we have a grating, I usually bring gratings and laser pointers, but since I'm so unused to speaking in front of people in real life, I didn't even manage to bring them, and it's kind of good because I almost got arrested for bringing a laser pointer on the plane once. But if we have a diffractive grating that's just a line grating, and we shoot a laser through it, we get all these spots. So what happens if we shoot an image through a line grating? We get all these images. So that's kind of cool. So now we've already gone pretty much towards this goal of these focal planes, right? But we can design functions that steer light almost any way we want. And what we wanted here was to fill a camera sensor. So we want to make 1, 2, 3, 9 images. So then we design a grating. We can etch these gratings in glass so that they're completely transparent, and we don't really lose a lot of light going through them, and there are open nanofabrication facilities to do this in. And when I sat and designed this, I didn't really know if this would actually work, and it was kind of scary, but it really did. So I wrote this algorithm. It's probably the worst code you will see in your life. Some people in the audience here have told me so, and they've rewritten it so that it runs in 5 minutes. It took 5 days for this to run when I ran it the first time, but it's perfect. Guys, you really get the perfect, theoretically optimal grading pattern. So what you do is you take a test pattern full of pixels, and then you flip one pixel in this pattern. Now you take the Fourier transform of each one of these, and you compare which one is closer to your target function of 9 dots, which, when we shoot an image through, is going to be 9 images. And then you just run that. And when that converges, you get your pattern. So this is a binary phase grading, so you etch this into glass, and the phase shift is pi for this little pattern guy, and then you get that grading. And here you can see the pixel flipper running, and it's flipping the pixels, and you get those 9 diffraction orders. So this is freely available online if you want to run the worst code ever written, or if you want to rewrite it. And you can also add levels. So I was talking to the guys in the nanofab, which was, they were telling me, well, but you can just add layers, right? And then you can make this pattern more complex. And it turns out you can actually make an 89% photon count efficiency grading, which is really important in microscopy, right? Because every photon is sacred. So if you do that, you get 89% photon count efficiency here. And it takes a long time to do that. I wanted to acknowledge some of the people I worked in the fab with. 2 of them here are even French people who came over to Cornell Nanofab to work in the lab with me. And it's really, really fun. So now we can multiplex the image. So we print our grading in glass, completely transparent. We put this pattern on it. That pattern makes it be 9 images distributed like this. So the first plus and minus diffraction orders in X and Y.
And then we wanted to focus shift it, because this was the whole key here. Like, we wanted to do 3D imaging. We don't want, I mean, you can also do things with multiplexing images. Maybe there's like a cool application for that too. And the thing about diffractive optics is that it's so fantastic that you can do that in the same device. So the period of the pattern and the wavelength determines the diffraction angle. So we have a fine pattern. It's going to mess more with our light. When it goes through, you're going to have a higher diffraction angle. If you have a coarser pattern, it's going to mess less with our light that's kind of trying to go through, and it's going to ripple less. It's going to have a smaller diffraction angle. So then we do some math, and we say, okay, if I take the Abbe-Sine condition, I have a point on the optical axis that's out of focus. I can calculate exactly the out of focus phase error for that point, and I can apply a distortion to this grading such that we get a focus shift. So what that looks like here is you have the 0th order that goes straight through, +1 order, -1 order, and here's for a periodic grading. Now we distort it. We get a higher angle where we have a finer period. We get a smaller angle when we have a smaller period. Smaller period, a higher angle. Everyone is with me. I'm just too jet lagged to speak properly here. I should make some students stand up and explain this. I'm sure they could do it better. But what happens to the beam then is that this beam here that's going into the +1 order is going to diverge, which means it's going to focus further away from the sensor than the regular focal plane. And the exact opposite thing is going to happen to the -1 order. Oops, that was very fast. So then that's going to have a different focal plane. So what that translates to in the image plane is that multiple focal planes from different positions are each going to end up in a different focus on the camera. So now we're home. We have our instant 3D imaging. We have not sacrificed any resolution. This element is highly photon count efficient, and we just pop it into the Fourier plane, and we get a 3D instant image. Does anyone have a problem with that? Anyone who's a physics person? Because there's a reason that nobody almost uses diffractive optics to image something, and that is Pink Floyd. Because they knew chromatic dispersion, and this is a prism. So this is just the refractive index change of the material. The effect of chromatic dispersion in diffraction is like 10 times stronger. So if you put green light through this grating, typical GFP fluorophore, say that you have a 30 nanometer wide filter, 500 to 530 nanometer, you get a green rainbow out. So this really, really doesn't work. And you get chromatic dispersion, and this is what that looks like in our microscope. Because of course, when I draw a microscope, I draw 2 lenses, the objective with a shorter focal length, and the tube lens with a longer focal length. And here in my Fourier plane, the back focal plane of the objective, I put my grating. And now the 530 nanometer light is gonna go at a higher angle than the 500 nanometer light, and this green rainbow comes out, and on the screen it looks like the Star Wars opening sequence when everything is blurred out. So the first thought here was, okay, let's put another grating in that just reverses this dispersion. Nice, because we can make a blazed grating where each little feature looks like a tiny prism, and that'll steer all of our light into the -1 diffraction order with 95% efficiency. So we put that in there, and bam, everything is back exactly where we started. And that was not what we wanted either. So then we decided the most beautiful and complicated element that I have ever designed, which is this multifaceted prism. It looks like you should hang it in like the Yuletide tree. And then it works. So now even like the physics people would be like, okay, yeah, that actually works. Like this works across the whole visible spectrum. And we got our multifocus imaging. And this was really cool. And a bunch of people, including people from Maxime Dahan's group and David Greenwald's group and Robert Tejan's groups, came and tried this out at Janelia. And with fast image, we can get these 3D images at 100 hertz if we have enough light, right? Because 3D imaging takes a lot of light. And we can study diffusion of biomolecules, transcription, and we know that there is no ambiguity within our 3D focal stack. So in the biology lab, you want things to be very safe from biologists, so you build these nice boxes around the optics, And that's also nice for the biologists, because if they see this, they're like, this is not a microscope. A microscope is white, and it has black knobs, and it has an eyepiece, and it has microfluidic devices, and it has all this nice stuff, right? So this is what a user-friendly microscope looks like. And thanks to software like Micromanager, you can also really easily control it, even though you have hooked up like whatever lasers or cameras or microfluidics devices. And there's some examples here of data. This is yeast centromeres floating around in a Saccharomyces, not pombe, which is the beer yeast, but the other one. It's called the Saccharomyces cerevisiae. Yes, nice. I get a star. I got a biology thing right. And if you visualize that in 3D, it looks amazing, right? So 3D visualization is one of those things that I'm really encouraging my students to work on, because it makes things look really wonderful. But the biologists, when they've used this for a while, they actually don't want it. They want the raw data, because they learn to see the different focal planes, and like they have this thing. Humans are amazing at image recognition. I saw some really nice workshops today on like deep learning, and it's really doing what we are doing. It's just throwing every algorithm that exists onto what we are looking at, right? So transcription is really cool, and then I got into this time of my life which was really awful, when my PhD advisor got a brain tumor, and the lab just disappeared, and Corey Bargman at Rockefeller University stepped in and just rescued me out of that situation and took me to New York and put me up with a place to live and a lab. But she was doing C. elegans imaging. And that's when I realized the biology is insane, because you guys label neurons functionally so that they light up when they are active. And you can even do crazier things, like people are doing optogenetics. But from my perspective, that's laser mind control, right? So you guys are amazing. And what I try to do here is, that gives me a great excuse to build imaging systems. So they put these little guys in olfactory stimulation, and when they smell almonds, unless they have COVID, because maybe they can't smell the almonds then, right? So they smell something attractive or offensive, and their neurons light up and fire. And here again, here they have mirrored neurons, so the top ones are one on the left side, and the right ones, the bottom ones are on the right side. And this really allowed us to image many, many more neurons at the time than was possible before, which was terrible for the students, because they were used to manually tracking 2 neurons. And now they had to manually track 14 neurons. So then we started working with Liam Paninsky at Columbia University, who was doing the spatiotemporal correlation. And we could automate, or semi-automate, I should say, the image recognition process, so that the program could find the neurons and see what they were doing. And it's really, really crazy. So here the almond smell goes on. So this neuron gradually goes off. This neuron goes off completely and goes back on again. And I thought that this one is activated. So those of people who are ambitious enough that they actually are going to understand how the brain works in detail, this is kind of fun data for them. And these are the sensory input neurons. So they're coming directly from the nose. You can see with EM the connections that are coming in. So that was fun, but note from the biologist, but we only labeled these neurons. We want to image all the neurons. This is all the neurons. Like, okay, what are you going to do with your 9 focal planes? Maybe not so much. So then I thought, okay, we can just build a microscope that has 25 planes. And then we did that. But that was like not the best design. And the reason that this is so small is not that the monitor doesn't fit it. It's really the field of view, because that's on one camera. And even if the camera is kind of big, it doesn't really fit. So I got the idea that, we should just have many cameras next to each other. And finally now that became possible, because now we have access to these machine vision cameras. They almost all have Sony IMX chips inside them. So it's CMOS chips. And I don't know, probably a lot of you guys use these in your labs now, because they are maybe not as good as a regular scientific camera, which has all these electronics and cooling that makes it 10 times bigger and more expensive, but it's actually good enough for some applications, and it's definitely much, much cheaper. And the optical design, as you can see, is much, much easier too, because that beautiful prism, you actually don't have to make it. You can just put the lens in front of each camera and a little grating, and you're home. So I didn't build that system, because then I got a job in California where I could hire students who could do it for me. And I'm so glad for that, because without that guy, how do you read out 100 frames per second from 25 cameras? And preview that on your screen so you don't have to like push a button. 10 seconds later, you can see all the images. Okay, I gotta adjust my focus, right? But this has been amazing. So I'm so glad I got this chance to do this. So Eduardo Hirata here, he built the system, and Anton has been working on the whole hardware control and image processing. And this was the first movie we got, and it's so bad. So Ed actually presented this at Focus on Microscopy in London before the pandemic. And you see the cameras start to fail too, right? So they just drop out. It's like someone's shooting them out. But this is the first time you get data on the new microscope. So just, guys who are building stuff, like this is how it's going to be. So thankfully later on, you get these awesome biological collaborators who give you beautiful samples, and you've somehow managed to get the system to work. So here's Jennifer Morgan and Gal Haspel from the Marine Biological Laboratory at Woods Hole, which is a fantastic place. If you haven't taken one of their summer courses, go and do it. You're not going to sleep for 12 weeks, but you're going to have the time of your life. And we also managed to get a video before the pandemic hit. So this is C. elegans with GCamp in the muscle. So they study spinal cord and movement. So the previous image that was the still image was a lamprey spinal cord, because they can regenerate their spinal cord. And this is then the undulatory movement of C. elegans of their muscle control. So that was really exciting. And then the pandemic hit, and we were not allowed to go to the MBL. So basically my lab has been dead for a year until this September, when UC Santa Cruz decided that we were allowed to travel again, allowed to go to our own lab again. I was allowed to be in my office again, and I was really glad of that. And we've launched these systems in the imaging facility, which is now open again. And we have happy users. Even though we opened the lid here, Rob was still okay, but you see he looks a little bit uncomfortable. And then how are we doing on time here? Usually I talk so fast I don't have to worry about going over time, but I have 10 more minutes, yeah. So now we get to super resolution microscopy. And we already know that we can get Ångström resolution with EM. But in EM, the sample is dead. And we don't want the sample to be dead. We want the sample to be alive. We want to look at it and what it's doing and its dynamics. So this is one of the solutions to that. There are fantastic methods. There's the whole localization microscopy field. There is STED. There is also like, is there any other really super resolution microscope method? Maybe not so much. The area scan I would still count as super S almost. It's going to almost 2 times. But structural elimination microscopy goes beyond this classical limit by extending the resolution by a factor of 2. And we can, for example, see the synaptonemal complex in this mouse spermatocyte spread. So we see the 2 sister strands 100 nanometers separated, wrapping around each other during cell division. This is oosex cell division, because it's meiosis. And I think it's fantastic, and it's incredible, and it's so beautiful. And SIM also improves 3D resolution and contrast. So it's not only the resolution in 2D. If you do 3D SIM, you actually get the 3D resolution, and this contrast improvement can be really, really, really helpful. And how that does work? That works by spatial frequency mixing. So if we talked about the diffraction gratings before and how fine the details spread light more, that's exactly the same as image formation. If you have a sample that has very fine features, those are going to diffract very strong, and your lens, unless it has an insane like NA following the, like covering the whole space, looking for every direction of the cell, it's not going to capture that fine information. So we can't get it back, right? When we do localization, we're not actually resolving things. We're saying, okay, I got a spot here, I'm going to deconvolve that. I know that the center of the spot is going to be in the center, like the location of the fluorophore is going to be in the center. But here we are tricking the imaging system by frequency mixing, spatial frequency mixing. So if you squint and you look at this pattern, you can't see it. If you squint and you look at this pattern, you can't see it. But you can see this moiré fringes, this mixed frequency. And since we knew what pattern we put on there, we can retrieve it back. So if we look at that in Fourier space, which is more intuitive, we see that we've actually improved our resolution here. So this is the aperture size, meaning the optical transfer function of the regular microscope. And here is the reassigned data where we have computed back out the underlying frequency that mixed with our frequency, and we've extended the resolution support 2 times. So that's really, really beautiful. And Here we have another little thing, which is the image reconstruction. So Marcel Müller was a PhD student when he wrote this free open source sim reconstruction software, which is really, really changed to me how I look at sim and how it is to apply sim, because it used to be that this software was locked up in commercial systems, and now you can just download it to ImageJ, and any one of you can download a sim dataset and you can reconstruct it. So that's really, really changing the world by free open source software. So again, the sim acquisition sequence is these stripes that we put on the sample. We have to put them on in several directions and angles, and then we have to slide them across the sample in order for these frequencies to mix in a way that we can retrieve them and extend the resolution. And when we do that, we can image beautiful things like chromatin structure. Here's a nuclear lamina, nuclear pore complex by Lothar Schermerle in Oxford. And we can, if we do nonlinear sim, which I'm not going to talk about today, we can go beyond this 2 times resolution extension down to 50 nanometers. Hesper Riego, who is a superhero in microscopy, even did this in a cell, even though it was a dead cell. So it's really impressive. But again, back to this temporal resolution thing. We can put this pattern on pretty fast. We can use a spatial light modulator. We can get these image frame sets at 25 hertz maybe, but that's for 2D. So a single focal plane of sim requires 15 images. Okay, 15 images, we can take them pretty fast. So let's say we want to take 9 focal planes of 3D sim. Now we're going to need 135 images. That is a little bit more challenging. So what we do now is we take our multi-focus trick and we just put that onto the sim. So we make time stand still in 3D, so every 3D frame really just comes, and then we image it over time. So then we're back to our 15 frames. So I thought that was a great idea, and I designed this system, and I thought, okay, let's build this. And then I got permission to prototype it in Stockholm by Hjalmar Brismar at funding from Stiftelsen för strategisk forskning. And unfortunately, since we built this on a commercial system that was really slow, things went too slow, even with like the 2D acquisition speed for us to be able to reconstruct it. So this is just a proof of concept. And now finally in Santa Cruz, thanks to my biological collaborators, who are really amazing people, we got a grant from the NSF to build this system. And as one of my students pointed out, that is always how it is. It is almost done. So things are almost working, and we're building it, and we're making it really beautiful. And I'm really hoping that together with those of these students who are still in the lab and haven't graduated yet, we will be able to present this at Focus on Microscopy in Porto, in Portugal, where I hope that most of you will show up too, because this is another one of those fantastic meetings. That was all I had, and I'm hoping that, yeah, that was pretty good timing, I think, maybe a little fast, but that means that there's a lot of room for questions.
Thank you.
Should we switch between them?
Maybe.
Or can you?
I don't know how to switch it on. Hello? Yeah,
got it.
Perfect. No, that was this one.
No, what?
Hello? Yeah, sorry. Thank you, Sarah, for this great talk. Do we have questions from the audience?
Now we hope that Max is really good.
Okay, yeah, I have actually a question.
Nice.
Yes.
That's what you're supposed to do.
Yes.
This one is not working.
Yeah. Okay. Since you really can do like imaging, like image like several focal planes like really at the same time, and like not compared to like other like 3D images to do it sequentially, can you? I mean, it's a very biological question, but can you think of any biosystem which really which you can image, or any biological processes which are like maybe fast?
The ones that we thought of so far were really like this neural imaging, functional neuronal imaging, where we want to image at about 100 frames per second, which is basically our camera frame rate at this point. And also transcription is Okay, and also transcription, of course, is really cool because we are looking at diffusing biomolecules, and that's one of those things where you can do multicolor, you can try to label some structures, you can see the biomolecules diffusing between them. This is really a question for biologists, I think, but what some people say that they want to do with this system, when I finally have gotten it to work in the imaging facility, is synaptonemal complex, and looking at multiple parts of the synaptonemal complex and the scaffolding, and like the dream would be if we could do this live in 3D, right? It would be so cool. And then, you know, if I survive as a professor, which is kind of a hard life, then maybe we can do that with nonlinear sim to really get the resolution in all the 3 dimensions. I don't know if it's possible to do that live. It depends on how bright your dice are, and if you can get this nonlinearity introduced either with saturation or bleaching or photo switching or who knows what, but that will be very challenging. But my dream data sets now are the synaptonemal complex things, where you can really see these strands of the chromosomes and then the scaffolding proteins between them. That's kind of right now what I am most excited about, but I'm also excited about all of these like other transcription things that come onto the microscope. You're doing an awesome job moderating here. Like, that's exactly what you do when nobody asks a question. Is there anyone? Oh, nice. We got one more question.
So I have 2 questions. So how much in the end light is stolen in the system compared to, like, you know, your regular microscope? And the second question, so when you do in the end, when you're trying to do sim with multiplane, so then the striped illumination also should be like 3D, right? So how do you arrange it? Is it like a separate illumination part, or is it still goes through the optics? Because of course in classical microscope everything goes up and down, and here, yeah, you need to have like holographic or something like that, yeah.
Yeah, so for the light efficiency, everything just multiplies. It doesn't just add, it multiplies. It's like risks. So when you have the multifocus grading, you're going to lose 10% there. You're going to lose another 10% in the first object of the chromatic correction module. You're going to lose a little bit in the prism. You have a relay lens system that loses some light. You're probably losing half of your light. In addition to that, you're doing 3D. So even before that, like compared to 2D, every focal plane is another exposure. So it's pretty grim. But I don't know many systems that don't lose half of the light. It's kind of basic. So, and in order to do that, we have really squeezed this. Like the multi-layer optics to make those in the nanofab, it takes you know, a couple of weeks of pretty hard work of etching in multiple layers using reactive ion etching plasma. So that's about where we get. And for the 3D sim, the 3D sim illumination pattern is modulated in 3D as well. This reconstruction is hell. Like, we still I'm still not completely convinced that we can solve this without moving the sample. But I think it should be possible because we do have modulation in each 3D point of the sample. But that will be a major challenge, and I'm actually bringing one of the students from Stockholm over who's really good at data processing, Maximilian Senftleben, and he's actually from Germany, but he's in Stockholm now. So in December now, in Santa Cruz, we're gonna have a hackathon. So if anyone happens to be around Santa Cruz, come to the lab. We got a lab dog, we got a lot of good coffee. We're gonna be sitting there in front of our computers doing Fourier transforms for 4 weeks. And I hope we can fix this.
Any more questions?
Okay, I'm not physicist, so I think I will ask a stupid question. But I try to understand why you don't try to do Palm and Storm with this approach. Is it possible to detect single molecules with this technique? Because I understand these small cameras, they will probably not sensitive enough for doing single molecule as CMOS cameras. But what are the limitations for doing Palm and Storm?
So we did. I didn't include the data, and one of the amazing things about that was that I got to go to the Riviera. So there was a group with Marcel Nolman at the CNRF, and they were looking at bacteria complexes. So we took a multifocus system there. His technician, Jean-Bernard Fisch, was like super magic guy, and then Laura Ojedi was also part of this project, and this was part of our PhD thesis. And when we did the localization in Z also. So we destroy our PSF in order to make it localizable in Z. So the way we did this was astigmatism. So astigmatism, you have a focus, you have a round spot, you have above focus, you have a line, and you have below focus, you have a line in the other direction. And the way to introduce this is you take a cylindrical lens, take some tape, you tape it onto your camera, and you're done. And then the calibration is the hard part, like the software, like to calibrate how deep in the sample do you get, how much elongation of your PSF in which direction. So we did that, and they published it too, and it's bacterial complex something segregate somewhere, biology. And I'd love for more people to do it.
Yeah, and another question is about, so you were talking about your fighting with this chromatic aberration problems and dispersion. Well, as I said, I'm not physicist, so yeah. So the question is, when you do multicolor imaging, colocalization of two colors while like 100 nanometer difference, how they colocalize well in different planes. So yeah, could you comment on that?
Yeah, so that's why we put this insane work into perfecting these optics. So this, the blazed grating and the prism together, or in the M25 system, just the two gratings, it completely removes the chromatic dispersion if you do it right. So there is no more chromatic dispersion. And I was, so then what you do is somewhere here, or even maybe before here, is the best. The best is to put the dichroic mirrors here. And then you have, the most I made was 3 arms, a blue arm, a red arm, a green arm. And then you build the whole system here in each arm, and you have a camera on each. And when I did this was a few years ago, so even getting all the cameras to work was also hard there, or maybe I'm just really bad with computers. But it was beautiful. And what you do is, just like when you do a focal stack in a microscope, you can have issues. The objective may not be perfectly corrected. So you have to calibrate it really, really carefully. So we put each camera on a little micrometer stage. They are made in Bordeaux. They are in the Newport steel stages. They make the best stages, precision machinery. So then you adjust your camera so that you have exactly the same focal plane for red, blue, and green. And you do that by putting a multi-labeled bead. So the bead will have all the 3 colors, and you can put a bunch of them. And you look really careful, and if you get a difference in magnification, you have a problem with your objective. So the objectives we used for that imaging were fantastic water immersion lenses from Nikon. It was called the VC series, and they had the correction color for the spherical aberration. It was beautiful.
More questions?
Hi, for the multifocus SIM, I was wondering, is it possible to use only one camera? Is there any issue with the number of pixels, the size of the pixel, to have enough sampling on each image for the SIM reconstruction? Or?
Yeah, so we're building that system with 1 camera per color, because those cameras need to be sensitive, like someone else was pointing out. These Sony chips, without all the cooling and all the nice readout electronics, they're not as good as the big, clunky, expensive cameras made for biological imaging. So we are putting several of those on the table. And actually right now, my students in Santa Cruz are comparing the back-illuminated CMOS to the EMCCDs and trying to figure out which ones we should be using. And the pixels are giant, so we put a relay mag in here, because for the SIM, we want to use kind of a lower mag objective, like maybe not 150x, which the pupil is this big. We're going to have to aim our laser beams in there. So we have like a 60x objective, which has an 8.4 millimeter pupil. And then we have a relay system here that we have to have anyway in order to form an image of the Fourier plane. And then we have a longer lens at the back so that we perfectly adjust the sampling. I think we're going to go with maybe 65 nanometer pixel size. Is that a good? Anyone else? Like maybe a little bigger, maybe a little smaller? I don't know. I think it's going to be good.
Okay, I think we are running out of time. So let's... there is one more question.
You can also ask questions over dinner, because I only know maybe 5 people here. So.
Oh, maybe one more. We have time for one quick question. Maybe you can.
Good engineering.
Thank you, Sarah, for your wonderful talk. It's me.
There you are. Hello.
I'm hidden. I'm sorry. I'm short.
And you had a mask too, so I don't even know.
All right. I just had a question on the last lens that is right before your camera. You were attacking it with like rays that have like quite an important angle and that are like on the edges, I guess, on your lens. Aren't you suffering from like quite important aberrations?
So this element really has to be aligned fantastically. So what we're using here is a 2-inch achromatic doublet. And we actually have been talking the last week about maybe we should use a photography lens instead, because they are really, really well corrected. It turns out that there's so many elements in them and so much glass and so many surfaces that we lose too much light. So I did a Zmax simulation of this. And the thing here, like this is already the intermediate image. So this is already magnified 60 times. So what I'm re-imaging with this relay system is not the sample, it's the image. So it's already magnified. And I did the simulation of this. If you have like even less than a millimeter misalignment of this lens, everything looks like hell. So it's really, really, really finicky. But if you put it in the perfect place, you have diffraction limited resolution.
Right.
Yeah. And the reason we crossed them over is they crossed over before, kind of. So it's kind of symmetrical and...
Okay. Gets along. Thank you very much.
Okay, let's thank Sarah again for this great talk.
This one works. This here. Where do you want me to put the mic? Ah, okay.
Do you hear me?
Monte le son du jaune s'il te plaît. Tu peux pas? T'es au taquet? Est-ce que vous m'entendez? Bonjour.
Hi, do you hear me?
Le son du PC, il faut que tu l'allumes. Il est allumé là? Alors tu peux mettre de la musique pour tester juste. On va balancer une musique pour voir si le son il sort vraiment. On vous remet le teaser. Vous allez le voir souvent. Ah non. Donc là, le son sort pas. Tu devrais recevoir quelque chose. C'est normal, Hugo, il est débranché sur ici. Il faut que tu viennes le brancher, je sais pas où est-ce qu'il se met. Sur quelle piste tu es? Non, non, ça va, c'est juste... Je sais pas où est-ce qu'il est. tu me dis. Je change? Ok. Vas-y. Non? Ok.
Can you hear me now?
On est d'accord, on est bon.
Okay. Okay.
So, hello everyone. Hope that you have a little energy left. So, allow me to introduce our last speaker for today, Nathan Shanner. With him, we will conclude the first module of the fluorescent sounds. He is specialist in biological fluorescence, luminescence, and optogenetics in the Department of Neuroscience at the University of California, San Diego, where he occupies the position of associated professor and where he dives to discover new fluorescent proteins. His researches are based on engineering naturally occurring fluorescent proteins and use them as biological research tools. So in video from San Diego, California, he will explain today how to build the next generation of genetically encoded probes and actuators. The floor is yours.
All right, thank you. Everyone can hear me okay? Yes? Okay. Yes. Thanks for inviting me. Sorry I couldn't be there in person, but I'm going to talk today about what my group does in engineering the next generation of better probes and optogenetic actuators. So since this is a session on fluorescent probes, I'll mostly focus on fluorescence, but I will talk a little bit about optogenetics and bioluminescence and how we're bringing all of those things together. So as a group, we make better probes, and that's our main focus, although we do some other things. And the strategy that we apply to this is to combine an engineering approach with an exploration approach, meaning we go out into nature and look for new materials, as well as engineering those things into probes that are useful. So to start, I'll just talk briefly about how we do the engineering. So here's some crystals made by our great collaborators at CNRS. And this is just an example of the kind of things we do. So these are 3 different fluorescent proteins that we isolated from corals. And now we have crystal structures of them, which is a critical part in sort of understanding what's going on with these things. And I think my video is probably not great, but as you've probably seen many times before, this is the structure of a fluorescent protein. They all look pretty much the same. They're an 11-stranded beta barrel. They're all about the same length. The structure is very conserved, in fact, conserved even into mammals. We still have this superfamily of proteins represented in our genome and as part of our connective tissue, actually. And they all have a central chromophore that's formed from a tripeptide that is, in nature, almost always tyrosine glycine as the second 2 positions, and then the first position is highly variable. And this chromophore forms autocatalytically, meaning that it doesn't need any external things except for molecular oxygen in order to be formed. So we use this process that you've probably seen a lot, especially in the last few years since Francis Arnold won the Nobel Prize, called directed evolution, to make improved probes. So to start, we take a wild type fluorescent protein, excuse me, or one that is not optimal for what we need. Then if we have a structure, we may start with some structure-guided design to make improvements, but often we just start with random mutagenesis, which means just error-prone PCR to introduce random mutations into the genes, then transform that into E. coli, and when you illuminate, then each colony has a single clone from the library that has random mutations. You pick the ones that are the improved ones based on what you're selecting for, which could be brightness, but could be other things like wavelength or photo switching or other things like that. Sequence those and then use those sequence positions that you've identified through random mutagenesis to then make more saturated site-directed libraries at multiple positions. And this generally gives you something that's much better than what you would get from random mutagenesis because you've combined multiple sites that cause improvements or changes in the chromophore environment or changes in folding or other things like that. So you select the best clones from these site-directed libraries, and then you just feed them back into this cycle where then you do random mutagenesis to identify new sites that might cause improvements and then continue the cycle until you've gotten things to the point where they're as good as they're going to get or as good as you need them to be. And of course, there's not really a defined endpoint to this process, so we always have to make a decision of where to stop because you can always kind of continue trying to improve things. So one classic example of directed evolution to improve a probe is Robert Campbell's work on monomerizing DS-Red back in the late 90s. And what happened with this was that what we do is to break the interfaces, so there's two symmetrical interfaces in this tetramer, introduce positive charges into the side chains in the interface to make it energetically unfavorable for the dimer to form. And then this usually does lots of terrible things to the protein. So then you have to go through the directed evolution process to rescue the fluorescence and make it bright and useful again. And so 17 mutations from wild type gave the first dimeric red fluorescent protein that was produced, dimer 2. And then a total of 33 mutations from wild type produced MRFP1, which is the first monomeric red fluorescent protein. Now, unfortunately, this MRFP1 had some problems. One of them was very low photostability, another was poor maturation. So as part of my PhD work long ago now, I improved MRP1 through directed evolution. I produced M-cherry, which most people are aware of and probably know about M-apple, but there's also a whole bunch of other color variants that we made that basically span the entire visible spectrum, or most of it. So by about 15 years ago, this is the collection of fluorescent proteins that we had available to us, which was a nice improvement over the original set, which was just these, pretty much these 4 on the left-hand side. Now, on the other hand, in modern times, we have so many fluorescent protein variants that nobody can really keep track very well. But we do have a really good database now, fpbase.org. If you're not aware of it, you probably are, but worth checking out. Has data on, well, the published data may not be accurate, of course, for some proteins, but the published data on pretty much every fluorescent protein that's ever been produced. And as you can see, for example, from the original aquariid GFP, AVGFP, this is the the tree of variants that has been produced from that are on FP base. So you can imagine that this is quite a lot to sort out. And that's part of the big problems that people have with trying to figure out what probes to use for their experiments, because there's thousands of choices now. We also make bioluminescent probes in the lab. I'm only going to just show this picture because I'm going to focus mostly on fluorescence for now. But we use FRET from bioluminescent proteins to fluorescent proteins as a way to improve the quantum yield of emission and also to alter the color. And it's a way to help evolve brighter probes. So if anyone would like to talk to me about that, feel free to get in contact with me. So I'll talk now about the way that we actually find new fluorescent proteins and new luciferases and other things that we're interested in, and this through exploration of the natural world. So this is a sort of general overview of the way that we actually get new fluorescent proteins from reef invertebrates. So it all starts by going into the water. This is at Heron Island in the Great Barrier Reef. And there's me when I used to not have so much hair. This is my husband, Jerry, who works in the lab with me. And this is our collaborator, Anya, who is the world expert on corals, really, when you look at it. So we go into the water and we collect coral pieces, and usually they're much smaller than the one that Jerry's holding here, but because we only need maybe a cubic centimeter of tissue really in order to get what we need. So we take these back to the lab on the island, and we first of all image everything. There's a nice fluorescence dissecting scope available, thank goodness. Not perfect, but very cool. And so here, this is just the polyp, one polyp on a coral called Pavona. And you can actually see individual cells here expressing a cyan fluorescent protein in the tentacles. And then along the sides, there's red fluorescent proteins. There's also red fluorescence from the symbiotic algae. And then there's green fluorescent proteins expressed in a different set of cells. So we always find this really interesting. The localization of the fluorescent proteins in the corals is so variable and really amazing to look at. So once we've imaged, then we can actually measure the emission spectra exactly using a mini spectrometer and a few little hacks to the microscopes available there and some postdoc and grad student effort. And what we get are things like this. So for this coral, these are the spectra that we got with different excitations. So for example, it has a cyan that seems to have a peak around 496. It has a green, very obvious green at 516, and then here we have the red fluorescence, which has a max around 650. And the red's always a little bit difficult because we get these extra peaks from chlorophylls and other pigments, but they're usually so far red shifted that we kind of know that those aren't real fluorescent proteins. So next we extract the RNA from the tissue. All the previous stuff is all done in living coral. So now we take a small piece of the coral and we extract the RNA. We do basic QC with a gel on site, which is tricky because if you've worked with RNA from mice or something, you would know that like there's a specific band pattern and ratio that you're looking for the 16S and 28S RNA fragments, but in the case of random invertebrates, this can be totally different. But we get an idea anyway. Next step is to actually take that RNA back to our lab in San Diego and then get it sequenced. So we've been doing Illumina in the past. We'll probably move to PacBio in the future. Then we assemble those into a de novo transcriptome for each sample that we've sequenced. And then, luckily for fluorescent proteins, they are homologous enough that we can just use a blast search against the de novo transcriptome to identify the putative transcripts that are fluorescent proteins. And from there, we can either synthesize the genes or we can just amplify them from cDNA. And then we clone them into our favorite E. coli expression plasmid. And then, well, here's one example of the diversity of fluorescent protein localizations and patterns in different corals. I always show this just to kind of get everyone excited about the natural world again. Sometimes you forget. Once they're in E. coli, though, we can actually then just purify them, the recombinant protein, and then characterize and evaluate them for their potential for becoming useful probes. Of course, the majority of fluorescent proteins that we find are very similar to existing ones. They may not really have any advantages, but there's always an interesting one in there somewhere. So this leads me to talk about our latest fluorescent probes. So about a couple years ago, well, when we actually discovered these new proteins, it was longer than that, but it was published last year, I guess. So we've discovered that Aquaria victoria, which is the jellyfish, of course, that yielded the first GFP that was cloned, we discovered its cousin, Aquaria australis, which is a known species, but it actually expressed a number of different fluorescent proteins. One of them was close homologue to AVGFP, but the others were quite different and looked really strange. And so we ended up going back and looking at Aquaria victoria, since we were able to actually get a sample of that from our local aquarium here at UCSD. And we found that Aquaria victoria actually does have a whole bunch of other fluorescent proteins as well. And so this is what we found. So in Aquaria victoria, we have the original AVGFP that was discovered, but then we also have this closest homologue, AVGFP1, which is actually quite a bit brighter than AVGFP, which is really funny, because if it had been cloned first, things may have been easier for everybody. It also expresses these three really interesting chromoproteins. So these these two have a 0 quantum yield. This one has a very low quantum yield green, but these two absorb in the green to red area of the spectrum. And they're interesting because they actually require blue light illumination to go from a sort of green chromophore to this far red absorbing, or not far red, but red absorbing chromophore. And in the Aquaria australis, we found the homologue of the AVGFP looks quite similar, but then we found this range of other proteins as well, 2 chromoproteins, which I'll talk about one of them in a minute, and then this photoswitchable chromoprotein, or sort of a cyan green emitting, but it's very low quantum yield. But it's actually a dry-clone type, so it's hydration of the chromophore that causes this shift, and it's reversible. But the one protein that's most interesting here is this ALSFP1, which has extremely narrow spectra compared to traditional AVGFP type proteins. And it also had a very high quantum yield, very high extinction coefficient. But it's dimeric, so we decided that this one was worth some effort in trying to monomerize. So here we have what we are about to preprint in the next couple months. This is what we're calling M-absinthe, and we call this way because the ABB, we originally had called the Aquaria australis Aquaria bluebell because it has blue pigmentation along the bell. So we kept that little abbreviation and just made a cute word, made a cute association. But so as you can see, we've retained most of the narrow spectra of ALSFP1. if you remember, I should have put probably a GFP spectrum as comparison, but GFP is much more over a sort of lumpy looking peak. It's much broader. The emission is of course narrower than the excitation on EGFP, but it still doesn't look like this. The 2-photon excitation is quite interesting for this too. It has very distinct 2 peaks. And the exciting thing for us is that this protein is at least 2-fold brighter than EGFP if you, depending on your illumination wavelength and your emission window that you're collecting. But in general, it's at least 2-fold brighter, even if it's suboptimally excited at 488. This is around here. And what we see in vitro so far, which usually correlates for us to the in vivo, is that we get actually equivalent photostability to EGFP versus neon green, our previous bright protein that we published several years ago, bleaches much faster. And we, because this is brighter, of course, there's a compensation thing where you get more photons, so you don't have to use as much illumination power. So it's not as big a difference as it appears here, where we're just plotting the brightness over time, not scaled to anything. But, so what we anticipate is that some of these clones are actually going to be more photostable effectively than MEGFP, which is something that no one's achieved so far with any protein, which has been a constant frustration for us, that MEGFP is still basically the most photostable green after all these years. But I think we finally fixed that problem. We also ended up making yellow variants. We can do this in a similar way to how you do this from GFP, the AVGFP. You put a tyrosine at position 203, which stacks on the chromophore, and that gives you a longer wavelength excitation and emission. In this scaffold, what we see is that we get sharper peaks for the YFPs, and we get some redshift relative to what AVGFP-based YFPs typically are. And we've managed to get the quantum yield nice and high. And so this is actually quite bright. It's, I can't remember how much brighter now than M Venus, but it's significantly brighter. And then the biggest advance is that we've managed to actually make the photostability basically equivalent to EGFP photostability in a yellow protein. So that should be helpful for a lot of people. And we also have found that these proteins, I don't have any photos here of the fusions in cells, but we've done quite a bit with this along the way as we were developing these, and these are the most monomeric proteins we've ever measured, so we feel pretty confident about that. So another protein that's really interesting from this Euphoria australis is this ASFP2. It's a blue chromoprotein. It's surprisingly, it expresses very well in E. coli. It folds and everything very well, so it's easy to measure. And we found it has a really strange looking absorbance spectrum. It has a pretty much symmetrical absorbance peak, which is very unusual to see. No discernible shoulder. I think it's just, if there is a shoulder, it's just sort of hidden under this broad main peak, so that it peaks around 610. And the crystal structure was a dimer. We're not sure in solution what it looks like. But the interesting part about this is that the chromophore is actually a completely different chromophore from what we've seen in any other protein. So what you have is a traditional GFP kind of chromophore here, but it's twisted in such a way that this is almost at 90 degrees to the rest of the chromophore. So it's totally non-planar, and it has this cysteine actually attached to the methylene bridge of the chromophore. So this is a completely different kind of situation than we're used to seeing. And so we're still investigating, what are the implications of this? Can this be made to be actually fluorescent? Can we get far red emission from it? And all of that is an interesting problem to have to work on. And the cool thing about this is that we've already shown very easily that if we take out that cysteine, we blue shift it back to absorbance of a GFP type chromophore. So the cysteine is absolutely required for this whole, this color change. And if we have mutations that probably make a smaller twist angle to the chromophore, then we get a blue shift as well, and then we get this purple looking colony. So the next new probe I want to talk about, sorry, I keep advancing too quickly, is called M. chelata. And this one is a variant of M. cherry that we made by essentially trying to remodel the chromophore environment to match that of M. neon green. And we succeeded in making something that is as bright basically as M. scarlet I in cells and is much more photostable. And so, and this is all measured in cells, so this is actual photostability in a microscopy context. So we believe, and we've done this several times, so we find that this is the most photostable red fluorescent protein we've measured. And we think that it's approaching the photostability of MEGFP now, so we're hoping that this will sort of rebalance the power dynamic between GFPs and RFPs in order to make RFPs actually more useful. Because everyone still has problems with RFPs either not being bright enough, not being photostable enough, or other issues. So we've fixed a lot of things with this one, and hopefully it'll be very useful to everybody. So we have more new FPs in development, of course. And now I'm going to switch a little bit to talk about bioluminescence, even though I know that's not technically the theme of this talk, just because it's too much fun not to talk about. And we do a lot of work on bioluminescence now, so I think it's something that needs to be on the radar for everybody in imaging. And there's advantages and disadvantages to bioluminescence, but there are some advantages that are worth thinking about. So when we go to the reef, we do find lots and lots of bioluminescent organisms. There's an amazing array of bioluminescent things in the ocean. So we have things like, this is not to scale at all, by the way, but ctenophores or comb jellies are classic bioluminescent organisms. They have photoproteins. Hydrozoans like this Clidia, this is Acoria, those have photoproteins as well, so they're calcium activated bioluminescence. There's dinoflagellates, which have their bioluminescence that is activated by acidification of their vacuoles. We have Odontocylus, which is a worm called fireworm, which has this beautiful green luminescence, and it's secreted and internal, and it's part of the mating display. And that one was very recently elucidated what the mechanism is. And the ostracods, which have a really bright secreted luminescence. So there's a lot of different things. Oh, and here's a nudibranch actually that we found that had luminescent gills of all things, and we still haven't figured out what's going on with that one. And I don't know how well this movie will go through, but there's native fireworms in San Diego, actually, in the bay. And if you know where to look, you can go find them. And this is one example. We took the lab out and we just dug some out of the sand, and this is how they look. They make this beautiful strobe kind of display, and here's a slow motion of it. Maybe will come through better than the other movies, but it's a pretty amazing bioluminescent display. And so we've cloned some luciferases from these local worms and from the ones that we found in Australia, but the luciferin is very difficult to work with, so we're sort of holding on that one, but who knows, we'll see what happens with that. So since I'm talking about bioluminescence, I should talk about what are the disadvantages of fluorescence imaging. Not that everybody here doesn't already know, but we imagine this is what happens when we're imaging a genetically tagged protein in a cell, that we illuminate it, and the only things that light up that we see are the tagged protein, right? But in reality, if you're looking at a tagged protein that's expressed at reasonable levels that, close to endogenous, often it's not that much protein. And you, of course, have all the autofluorescent molecules also present in the cell, which you can't avoid. In bioluminescence, of course, you don't have to worry about autofluorescence because you put the light source directly at the protein. And so this eliminates the need for external illumination and gets rid of vast majority of any background signal. And the price you pay, of course, is that the actual magnitude of the signal is going to be lower, although we're working on that and getting things much closer to parity with fluorescence. I won't talk about that today, but that is something that's in the process of going. So bioluminescence is light generated by enzymes by oxidation of a luciferin. Here it's shown as LH2. Usually it's molecular, other things. And then you get light and you get some byproduct and an oxidized luciferin. And so quantum yield in this case is just photons emitted over oxidized molecules. If you connect a fluorescent protein directly to a luciferase, what you can do is then, because the energy is transferred, if you can get it transferred efficiently, so if you get a very high FRET efficiency from the luciferase to the fluorescent protein, what you can do is have a fast luciferase that's oxidizing lots of luciferin molecules, but has a low quantum yield, which means most of those oxidations don't lead to photons being emitted. If that energy is all transferred to a high quantum yield fluorescent protein, then the fluorescent protein acts as if it was excited by any other source and emits light with that same quantum yield. So if this quantum yield is 80% and this quantum yield is 1%, but you have high FRET, then what you get is luminescence with an 80% quantum yield. And so this is the trick that we can use to kind of decouple, the catalytic rate of the enzyme while we're evolving it from the quantum yield, so that if these things are coupled normally, which we assume that they probably are to some degree, putting on the fluorescent protein actually decouples that, at least to some degree, because FRET efficiency also depends on the quantum yield of the donor. So there's limits to this, but we can actually probably make things better this way. And this is what the strategy that we do use. So beyond just making bioluminescent tools for imaging cells, we also use bioluminescence to control cells. So I'll just go over this really briefly. I think everyone has heard of optogenetics and has an idea of how it works, but it's a general term for being able to put something into cells that responds to light and creates some biochemical change. So the most common is these ion channels, channel rhodopsins, and the related channels that, when illuminated, are positive ion channels and can lead to depolarization of cells. And there's negative ion channels and proton pumps and lots of other things that do various other things to cells. You can make them inhibitory, excitatory, you can change other biochemical activities, gene expression, everything. So there's optogenetic tools for pretty much everything. And it just so happens that my colleague Ute Hochgeschwender, probably about now 10 years ago, or maybe a little bit more, noticed that the emission spectrum from Gaussian luciferase matches quite well with the activation spectrum of channel rhodopsin 2. And so if you couple those two things, then what you end up with is a luciferase plus a channel rhodopsin. So it's a luminescent channel rhodopsin, or we call luminopsin, or LMO. And so the original construct was Gaussian luciferase attach the N-terminus of channel rhodopsin that's then also tagged with YFP just to be able to see it. And what you end up with is something where when you add selenerzine, which is the luciferin for Gaussian luciferase, it generates light. There's fret from the luciferase to the channel, and that activates the channel. And so this is some early examples of excitatory and inhibitory luminopsins, and we call this whole approach BLOG, which is bioluminescent optogenetics. So in this case, this is an excitatory channel, so here's the localization of the luminescence, and when you add selenerzine, you see a massive increase in spiking. And then the rate of spikes seems to correlate pretty well with the bioluminescence output, which indicates how much of the substrate is left. And in this case, we have an inhibitory one, so the channel is inhibitory, same luciferase, and when you add the luciferin here, then you get silencing. And then once the luciferin is used up, then you get activity again. And this can be used in even a potential therapeutic way. In this case, this is a project from a grad student who's now a postdoc. And here, what they did was they had a spinal cord injury, and then distal to the injury, they injected AAV expressing this excitatory luminopsin. And then for a week, they get daily injections of selenerzine to stimulate the neurons that were transduced here. And what we saw was that actually the treatment led to a greater recovery from the injury, meaning that stimulating these neurons kept them alive probably and it allowed them to reconnect. And so this is another potential application for this kind of technology. The big advantage, and this is a pretty old slide now, but the big advantage of the BLOG system, this bioluminescent optogenetics, is that it's pretty much modular. So we could choose any number of luciferases as the inputs and any number of channels or other optogenetic elements as the outputs. And so by mixing and matching these things, we can create basically chemogenetic versions of any optogenetic channel, any optogenetic element. And just a quick plug for our collaborative group here. These tools are all freely available if anyone's interested in trying them out for their research. And we are happy and very excited to try to get people using these. And so here's our website for the collaborative thing. It has a lot of information about this technology. So now I'm going to switch gears one more time and talk about how we can take everything and combine it together to sense some really interesting things. So first of all, we're working on making much better luciferase-based calcium sensors. So everyone's probably familiar with sensors like GCamp. This is very similar in concept. We have a split luciferase, and we put a calmodulin and a calmodulin binding peptide in between the two parts, optimize the linkers, optimize lots of other things, and end up with something that is luminescent in the presence of calcium and dark without calcium. And if we do this right, what we want to see is that the contrast or the luminescence at resting calcium levels in the cells should be nearly zero. And so we've managed to actually do this. So our best clones at the moment, actually this is a bit old, we have somewhat better ones, but as you can see, the baseline is very low at, resting calcium, 50 to 100 nanomolar. There's basically no luminescence. And then when you get to, higher calcium, like 1 micromolar, which would be, 1 micromolar, which would be sort of, a close to the membrane during a depolarization kind of situation, then you get a lot of a lot of luminescence out. And surprising part for us is that the Hill coefficient's 7 for this, which doesn't really even make sense, but it is what it is because that's what we've observed. So there's a very sharp transition between on and off states in these sensors. And that's helpful for some things that we want to do. So we're calling this CABLAM, which is Calcium Bioluminescent Activity Monitor. So that's how I'll refer to it on the next couple slides. So if we look, say, in U2OS cells and we just stimulate with just calcium and ionomycin, so the substrate is present throughout. But before we inject calcium, it's essentially at 0. And then when we inject, then we get, you know, say, in this case, about a 30-fold change in luminescence. And this decay you see is likely due to either reuptake of calcium into the ER or just using up the substrate because we only put it in one time. It's not continuously flowed through here. If we try this in neurons with perfusion of the substrate and things to stimulate the cells, you see, first of all, it expresses nicely, and you can see it nicely in the soma, but also in the processes. And then with high concentrations of glutamate with luciferin, so this is luciferin alone added right here, and then with 100, 200, 400 millimolar glutamate, and then finally with ionomycin to max out the calcium signal. You can see that there is a dose-dependent response, a little bit weird, but these are very high concentrations of glutamate, so it's sort of what we might expect. If we go to low concentrations of glutamate, we actually find that we can detect signals from some cells at even 10 micromolar glutamate, which is a lot lower than what we'd see in the synapse. So we think that this is going to end up being useful. It's just a matter of getting all of the parameters for the imaging right. And imaging bioluminescence is definitely more challenging than fluorescence. You know, you can't control the plane that anything is, you know, you can't control the excitation at all. And so there's other complications for sure. So you can't do SIM with bioluminescence. If someone can figure that one out, that would be great. So that's the calcium sensor. Next part of this whole story is this ultra-sensitive photoconvertible FP, UPFP that we're calling it now. So what happens with this protein? This is a protein that we found from a reef invertebrate. It begins its life when it's expressed as a green fluorescent protein. See this top curve. This is what it looks like if it's never been exposed to light. And then with very small amounts of blue light, it photo switches into this other form. And this is reversible. If we illuminate with 400 nanometers, most of this comes back. But the interesting thing about this photo switch form is that it ends up with this weird long wavelength thing. And so we don't know what species this is. But what we do know is that it doesn't take a lot of photons to get there. Now, if we leave this overnight, or even a few hours, what we see is that there is a dose-dependent development of this red absorbance, and this actually ends up having a 650 nanometer emission peak with a 0.3 quantum yield, so it's pretty bright. And so what we get is that the amount of blue light that this protein saw can be read out as the amount of red fluorescence in the end. And because it takes so few photons to do this, compared to things like MEOS, which would take thousands of photons, that means that we can actually use bioluminescence FRET to convert this protein. So here's our first trial that we did several years ago now on whether or not we can actually see photoconversion using bioluminescence. So this is just a fusion of luciferase to this protein, luciferase, that emits blue light. And so if we don't give it any substrate and we keep it in the dark, we don't get any red fluorescence. This is just green excitation versus red excitation. It's a tetramer, so you do get fret. That's why we see this. And so the more substrate we use, this is a tenfold change, the more red fluorescence we get out in the end. And so, putting these two things together, the bioluminescent calcium sensor that has a very high contrast between resting and active calcium, and this ultra-sensitive photoconvertible fluorescent protein. If we connect those two things together, what we end up with is a memory dye that tells us how much calcium activity the cells have experienced while they had the luciferin substrate present. So when they could make light at high calcium, that light can convert the protein and turn it red. So here's our first check. And here's what we turned it off first. It turns off completely in the image in the far red channel. And you see that over a few hours, the red fluorescence develops only in the cell that you illuminated. So because this is so sensitive, this is how we have to work. We have our red LEDs covered with a red filter to make sure there's no blue photons at all coming out, because we found that even if you use a far red LED, that's still enough to convert this protein. And so if we put this is in U2OS cells. We're working on getting this in neurons. It's a little bit more tricky to get the construct into neurons because it's kind of big. But what we see is that if we express this in cells and we don't give any treatment and we don't give any luciferin, then we don't get any red fluorescence. If we just give the luciferin, we also basically don't get any red fluorescence. If we give the luciferin and ionomycin so that all the cells end up with high calcium, then we get strong production of red fluorescence that correlates with the remaining green. So we know this actually does work in cells. So this is showing that we can read out. It's essentially like if you know about Campari, that's very much a parallel kind of system, except it requires external illumination with 400 nanometer light to, instead of, basically we're substituting the supply of the luciferin with the 400 nanometer light that Campari requires. So this is much more gentle, and also could be accessed in a whole animal, in a whole brain, without being invasive. Because you can apply this, you can give this lenosine peripherally. So if we look at the results of this first trial that we did on this sensor, we see as we look at the far red intensity from green excitation, then we get a clear difference between the no treatment luciferin alone and calcium plus luciferin, ionomycin plus luciferin. And if we look at just the far red to green ratio, it's even tighter actually, we get pretty good. And then the interesting thing about this is the luciferin alone, if you look at these ratios, there's a couple of outliers that are really red. And this could indicate that that's actual biological activity, like maybe the cell did have a high calcium signaling event during that period. This is something we have to look at a lot more carefully. So I just want to make sure that everyone knows that if you'd like any of our probes, if you'd like to try them, this is why we're here. This is why my group exists, and why I like to do what I do, is to make things that are useful to people. So please get in touch with me if you'd like to try any of these things, or ask about anything else that we might have available to test out. So the things that we do have ready for beta testers, or maybe even, they're nearing final release, some of these things. So the ultramonomeric green and yellow variants of ALSFP1 that I talked about, the bright and photostable ultramonomeric red FP, the M chelata that I talked about, What we also have in the works, a nice monomeric orange FP derived from a new fungia species of coral. We have a photo-switchable green monomer that actually is dark by default, so it relaxes to a dark state. So it's really nice for highlighting, and we are going to be using it for some other interesting things. We have dark Fred acceptors, so we have a lot of low quantum yield monomer variants that could be useful for film and other stuff. We have photo-switchable dark acceptors, so that's an interesting one. They're derived from this photo-switchable green monomer. So these start off absorbing 400 nanometers and then can be switched to absorbing, around 480 to 500, but with a zero quantum yield. This could be useful for some application. We have some really interesting long Stokes-Shift red monomers now that we're going to be optimizing here, and we actually have a long Stokes-Shift green variant of ASFP1 that I didn't talk about, which is unique in that it has absolutely no excitation with blue light, so it's completely separable spectrally from a regular green. And then finally, what I didn't really talk about, but is a big thing that we're going to be publishing one soon, is bright soluble luciferases and luciferase FP fret pairs. So we have something that's 5 times brighter than nanoluc at this point. So that will be out soon. So I'll wrap up here and just say thanks to all of the people that, of course, went into doing all this work and all the funding as well. Been a lot. And as I reminded you, please take a look at our bioluminescencehub.org website. I think you'll find some interesting things. And please be in touch. And I'll leave this movie playing, and I can answer questions.
So, hello, yes, great talk, great work, thank you. Two questions about the calcium indicator. I may have missed it, about what is known about the kinetics, the speed.
So that's still in question, so we don't know exactly. Well, part of the problem for us has been that in cells it's hard because still getting a bright enough signal is difficult, so we don't know exactly what the kinetics are, like the on and off kinetics are. If we look at our data on our plate reader where we're injecting while reading, it looks pretty fast, but it's hard to tell because there's mixing effects and other things, so it's hard to know. So we'll have to do some more kind of serious biochemical experiments like Giacore or something like that to really get a good idea, I think. It's hard in cells because we can't image fast enough yet to really measure that. We're at best we can do 100 millisecond, 10 hertz kind of imaging with this sensor so far. But we're working on making it brighter so that will be easier.
Okay, and then what about you mentioned it has a very steep, very high Hill coefficient. So so it's like a detonator kind of thing then, right? Is there a way to also quantify the signals, to calibrate them?
Yeah, that's the other thing. Yeah, of course it's gonna be, there is a little bit of luminescence from baseline, but it's usually too hard to measure. So, at least when you're imaging, it's awfully hard to measure. And so, calibrating is going to be a bit of a trick. We've thought about, we could make it ratiometric by adding a second luciferase with a different color output. Or we could try to use fluorescence as some way to kind of normalize things. But we still have to figure out how to calibrate. If we care about calibrating, the question to me is whether or not it matters for most experiments. You can't really calibrate GCAM, like you kind of can, but you can't really. So maybe it doesn't matter that much, but I think that, yeah, we're working on that part about being able to calibrate, because the high contrast is good and bad because of that issue.
Great, thanks.
Mm-hmm.
Thank you for this nice talk. I wonder whether, when looking for these new fluorescent proteins, folding speed or folding efficiency is still a question. I mean, you discussed brightness and quantum efficiency, so I was wondering whether this is still a criterion or not.
So far, what we're seeing is like the green variants seem to be very efficient folding. We're measuring the brightness in cells with a T2A M-cherry, so we have an internal standard. So that gives us at least, a good measure of actual practical brightness, and that tells us a lot about how well they fold and mature. And we're working on getting like a good measure of maturation time. I'm personally not a big fan of just looking at oxygen-dependent maturation time, because that doesn't tell you a whole lot about folding or anything. So we're working on some cell assays where we, there's been a few groups who have done beautiful work on maturation time in fluorescent proteins, but they're not systems that I can replicate in my lab very easily. So we're working on making our own assays in mammalian cells. But the general feeling that I have right now, especially given the brightness in cells compared to existing FPs, is that these guys are maturing pretty well, folding about as fast as traditional FPs, so they're just a little brighter.
Thank you.
Another question?
Yeah, thanks. Great talk. I was wondering, so you made a great step forward in improving the photostability of house FP1, and I was wondering what's the mechanism behind? Is this related to like oxygen accessibility to the chromophore or something else? And also, do you see like place for further improvement of the photostability? Can we ever dream of reaching the photostability of an organic dye with an FP in the future?
This is a very difficult question. I mean, we don't have structures for our most current ones, so it's a little hard to say exactly what the mechanism is. We know that, there are some mutations that affect photostability that don't make any sense to us, that are too far from the chromophore to be directly doing anything, so they must be just rearranging hydrogen bond networks or something. And so we assume that part of it is that we're eliminating oxygen binding sites, so that, or we're limiting accessibility. We can't be limiting it so much the chromophore doesn't mature, of course. But so I think there is somewhat of a trade-off between maturation time and photostability, but we're not seeing a huge amount of that. So we still have not come up with any general rules about photostability and what kind of mutations affect photostability to actually be able to predict a whole lot. It's gonna still be very randomly oriented for a while. I think the way we approach photostability is gonna be, not very hypothesis based on some level. I mean, we know the positions that are important, and sort of, they're in general all around the chromophore, but we don't know the mechanisms. And I mean, there's very little research out there on actual mechanisms of photobleaching. If you look, to try to figure out, what are the actual things that are happening in the protein when it bleaches? What are the species that are formed? That's something that I would love to have funding to look into, but I think it would be awfully hard to get funding for. So, I think photostability is always going to be sort of at the forefront of probe engineering, I think, just because it's so critical. So eventually, I hope we do understand it a little bit better. But right now, I know that, say for MRFP to M-cherry, there was a group that did some molecular modeling studies showing that the mutation at position 163 had eliminated an oxygen binding site, and that's probably why it bleaches more slowly than MRP1. But that's sort of an anomaly in the literature. I think that there's an actual answer to why something's more photostable.
Okay, one last question, please.
Yes, one long-standing question is the biological relevance of fluorescence, and I wanted to know how you choose your corals, in fact. Do you proceed randomly, or do you know what you are looking for?
So, we know we're looking for red proteins most of the time. I mean, everything has green proteins, so we sort of, as far as knowing what we're looking for, it's it's sort of embarrassing, but we do sort of sample randomly. I mean, to some degree, because often what we find is that there's fluorescent proteins in these animals that are expressed at lower levels that we wouldn't even observe, but we can find the sequences, and if we clone them and express them, they're sometimes very interesting. So we've sort of learned to sample everything we see that looks like it has any fluorescence just in case, because it's cheap enough. The biological function of fluorescence is much more complicated, I think. I think it's probably multifunctional depending on the color and the organism and the location in the organism. There's been very few convincing studies about the function, although there's some evidence that corals that express higher levels of fluorescent protein may be more resistant to bleaching or more resilient after bleaching, may be more likely to survive bleaching. There's some kind of sunscreen sort of hypothesis, but it's very hard to prove these things. There's been some evidence that in some jellies use fluorescent proteins as basically a lure for prey. In, say, Aquaria victoria, it's more of a way of changing the emission wavelength of the luminescence. So, in Aquaria, the photoprotein is coupled to the GFP, and so you get green emission instead of red, instead of blue. And that's probably because of they live near the surface where there's lots of chlorophyll, and green light is actually more penetrant than blue light. But there's probably a lot more functions for biological fluorescence out there. I think it's one of those multi-purpose things that can be used for a lot of stuff, just like bioluminescence. It's used differently by different organisms. So hard to say, but we try to sample things that are, interesting and different, but we don't always know what we're going to get in the end. We often don't have any idea. We know there's certain species that I know that I want to get the fluorescent proteins from that we still haven't managed to do. But other than that, it's sort of surveying what's there and trying to find the most interesting looking things, or things that haven't been sequenced before.
Okay, thank you again, Nathan. Thank you for your presentation. It was so fluorescent.
Thank you. Thank you.
Thank you. Goodbye. Pas pour lui.
Thank you. Bye.